Hello, everyone. Welcome to another seminar. So today it's my absolute pleasure to introduce Allison Miller, who's coming to us from the St. Louis region. And so Allison did her PhD at WashU in St. Louis and also Missouri Botanic Gardens, where there was a really nice joint program there. She currently is the interim co-vice president for research at the Danforth Plant Science Center. And so you can talk to her about the interesting jobs of learning how to manage at big scales. She's also a professor in biology at St. Louis University and a research associate at the Missouri Botanic Garden. Another thing that she's currently doing is she is the director of the NSF BII for New Roots for Restoration. And so her research spans, ever since, we met, we started faculty jobs in St. Louis at about the same time. And so she was my go-to from a different institution. So across early facultyhood, we would meet weekly to talk about the exciting things that are happening in our lives at that time and how to manage the next step. So she's just one of my phone of friends, go-to people. And so ever since I've known her, she's always studied perennial crops and their wild relatives. And so this has taken her from places around the tropics, studying species in the Anacardiaceae and how those have been developed for agricultural locally, to horseradish, to pecans, which are the only native perennial tree that we eat nuts from. Here in the US, and then she really made it big with grapes, not surprisingly. So fun things like the French grape stock has grown on Missouri roots. And so she combines things like looking at the role of agriculture and selection, taking an evolutionary lens. She's at Danforth, so she has access to things like phenomics, and so really mass processing what things look like. Does a lot with root shoot connections and thinks about trait diversity. So it's just an absolute pleasure to have her here with us to tell us some of her story and what she's finding. So thanks and welcome, Allison.
Thanks, Amy. Well, good morning, everybody. Thank you very much for coming. Amy, thanks for the invitation. I've heard a lot about Carey Institute over the years, decades, but this is my first time here, and I'm excited to learn more about the Institute, talk with many of you about your work, and share some of what we're doing. So the title of my presentation today, Capturing the Perennial Plant Phenotype from Biological Foundations to Breeding Applications. I thought we would start by just looking at why and thinking about why I am motivated by what it is that perennial plants are actually doing. As we look around the world at different natural ecosystems, like this beautiful Pacific forest in Costa Rica, to places like the desert in Arizona, up to the Rocky Mountains, Rocky Mountain National Park, all of these breathtaking landscapes are full of long-lived perennial plants that are growing together in diverse mixtures. This is one of my favorite places, Pictured Rocks National Lakeshore up in the Upper Peninsula of Michigan. And even closer to home for me, the native North American prairie. This is about 35 miles southwest of St. Louis at the Shaw Nature Reserve. So one of the things that motivates me is understanding how long-lived plants grow, how they allocate their resources to different parts of their bodies, how they're responding to some of the environmental changes that we're seeing today, and also how they can be harnessed to both meet the needs of people, but also continue to provide these key ecosystem services that we know are at their core. So in addition to these basic biological questions that I'll run through today, I'm also thinking a lot about how long-lived or perennial plants can play more prominent roles in agriculture. Where I live in the Midwest in St. Louis, a lot of our landscape looks something like this, where we have annual plants grown in monocultures, mostly soybeans and maize. There are huge differences between annual monocultures and these diverse perennial systems in nature, not just in terms of plant composition and longevity, but in the impacts that these plants have on the soil and on the landscape. So there's a number of folks who are increasingly interested in opportunities for perennial polyculture, agriculture. And many of the questions that we're asking, these basic biological questions, have that kind of broader goal in mind. Okay, so research-wise, I run a lab group that's physically based at the Danforth Plant Science Center. As Amy said, the Danforth Center, like Carrie, is a private, nonprofit, independent research institution. It's about 25 years old, and the mission of the Danforth Center is to improve the human condition through plant science. My lab group at the Danforth Center is focusing on a number of different questions and systems, but basically they center around these bigger picture questions. What are the genetic and environmental determinants of perennial plant phenotypes? How do traits co-vary among organ systems, above and below ground, and how does that change over temporal scales, over time? And increasingly, how do perennial plant traits influence aspects of the soil structure, composition, and gas flux? This is a new area for my lab group, but I will say a little bit about it today. So in thinking about capturing the perennial plant phenotype, I work at an institution where a lot of folks are interested in plant phenotypes or plant traits, but a lot of those traits are being observed in short-lived annual plants. A lot of them are being observed in plants that are critical for agriculture, as I mentioned. And a lot kind of start at the organism level and move downwards into organs, tissues, cells, molecules, genetics, etc. And this kind of plant organismal biology approach to understanding phenotypic variation is focused on many traits, often really technologically advanced in impressive and exciting ways, but relatively few species and also ultimately often relatively few environments. This is one kind of body of work and approach to studying plant phenotypes. The other one is what we think of as the functional trait ecology literature, where folks are a little more interested in these kind of higher levels of biological organization, interspecific interactions, community level processes, even on up to the ecosystem. And often what I have noticed is those types of studies may focus on many different species and maybe many different environments, maybe totally different parts of the world, but focusing on relatively few traits and perhaps traits that are observed or gleaned from databases, maybe a relatively few individuals, lots of different, kind of just a different perspective on how to study plant biology. So we're trying to, and what I'll do today is start at the level of the organism to try to begin to capture what it means for perennial plants to express all the different forms that they might express in all their different organs over time. And what we'll do in this talk is then move a little bit in this direction, kind of towards the organ and tissue focus, and then ultimately a little bit in the other direction. This is very much work in progress, a lot of ongoing studies that haven't been published, but I wanted to take the opportunity to get this crowd thinking with me and helping me kind of frame some of the questions that we're addressing. So again, what I'll do today is focus primarily, at least from the first maybe 3/4 of this talk, on biological foundations. And I want to walk through 3 questions, which are just a bit of a snapshot of some of the projects that we have going on. The first, how do root systems modulate phenotypic variation above ground? How does this change over time? We'll look at an example from grapes to study that. Second, how do traits co-vary among organ systems, and does this change across temporal scales? Now, in this group of studies, we'll look at a few different species. We'll move from a woody system, which I'll introduce in my first point, to more herbaceous systems. And then lastly, how do perennial plant traits influence soil structure, composition, and gas flux? This is among the newest of things that we're currently exploring. I am not a soil scientist. I would like to be a soil scientist. I would like to work with soil scientists, but these are questions we're still figuring out how to frame, and I certainly welcome your questions and input. Okay, so first let's talk about root systems and how they modulate phenotypic variation above ground. And for this, we're going to spend a little time in our grapevine projects. This is work that's been and is funded by the Plant Genome Research Program at NSF. And for almost 10 years, we have been touting grapevines as an ideal model for understanding perennial plant phenotypes. Grapevines are among the oldest and some would say most popular woody domesticated crops. They're among the most economically important. I think they may be the most economically important berry crop in the world. They are clonally propagated. So you have the same genotype growing across huge swaths locally and even internationally. And they're also grafted. And so we can experimentally manipulate root-shoot interactions by grafting the same clonally propagated scion or above ground part of the plant with a different genetically distinct rootstock. All right, so this is a vineyard in La Rioja, Spain. I was at a meeting there last summer, highly recommended if you have a chance to visit this part of the world. This vineyard is full of the Vitis vinifera variety Verdejo. This is a white varietal commonly grown in this part of Spain. But what you might not know, or maybe some of you do, is that although all of the vines in this vineyard, as far as I remember, are Verdejo, the rootstocks are a different species, actually, a hybrid derivative of two North American native grapevine species, which are, as Amy mentioned, now used as the rootstocks, the root systems of global viticulture. I'm happy to talk more about the history of how this came to be. I'll just mention grafting is an ancient technique that dates back to the Romans. But in grapevines, it started in the mid-1800s following the introduction of an aphid named phylloxera from North America into Europe, where it began to decimate European vineyards from the root systems up. And the solution was that now most vineyards in most parts of the world consist of European grapevines varieties grafted to North American rootstocks. All right, so we've been using this system to try to understand how different root systems change what's going on above ground in plants. And I'll just tell you about a study we did a few years ago in an experimental vineyard at the University of Missouri's Southwest Research Station in Mount Vernon, Missouri. This is about 3.5 hours southwest of St. Louis. And in this experimental vineyard, a guy named Keith Striegler, who used to work at the University of Missouri, now is with E&J Gallo Winery, took a local varietal named Chamberson, and that's what all these kind of gray vines represent, grew it ungrafted, and then grafted it to 3 different rootstocks. Now, in the grapevine world, each one of these rootstocks with these funny names, like 1103P, is a clonal F1 hybrid of 2 North American rootstocks, or 2 North American grapevine species. And there are 3 species in total that are used, 2 of which made each one of these different crosses, or different hybrids. And these are like commercially widely used rootstocks. Probably if you're drinking wine from grafted vines here, they're almost certainly grafted to one of these rootstocks. So anyway, this set of 4 combinations where we have the same above ground part of the vine with different rootstocks was replicated 72 times in a randomized block experimental design with an irrigation treatment. So it was really fortuitous that we literally stumbled upon this vineyard and began collecting data in about 2015. The data I'll present for this study were collected in 2017, 2018, and 2019. And what we did for 3 years is we went into this Chamberson vineyard 3 times this season, at anthesis, when the flowers were just opening, at veraison, which is the time when grapevines turn from green to red, and at harvest. And at each of those time points over 3 years, we tried our best to comprehensively phenotype these Chamberson vines. So we collected data on leaf shape, vine physiology, the elemental profile of the leaves. We did transcriptomics both on the leaves and the flowers and the fruits. We had an enologist on our study who looked at berry chemistry and wine chemistry. There was a graduate student who looked at the microbiome of these different grafted plants, and then another graduate student who studied the epigenetics in the shoot system associated with these different rootstock and irrigation treatments. Now, I'm only going to talk about a few of these types of data today. The take home is that the rootstock affects, albeit in a subtle way at times, every aspect of the functioning of the scion. So what goes on below ground really matters for the traits that we see above ground. So I'll just give a couple of examples. I'm going to start first with the ion concentrations in leaves. So of course, the root systems are the way by which elements enter into the plant. They then move up the plant and move into different leaves and different parts of the above ground part of the plant body. Zach Harris, who was a former PhD student in my lab group, then a postdoc. He's now a research scientist at St. Louis University. He did all of this work, did a wonderful job with it, and basically showed when we look at different elements, here I'm showing magnesium and molybdenum in the ungrafted plants shown in gray, and then the plants that were grafted shown in green, purple, and yellow. There are differences in the concentrations of these ions in the leaves of Chamberson, depending on what the root system is. Now, what's really interesting is that those differences change based on a number of different factors. So in the first graphs up here, we're looking at the year. So we can see that the concentrations, again, this is just an example, molybdenum and selenium, the concentrations of those ions differ in these grafted vines based on what year we're looking at. So there's a rootstock by year effect. We also can see, looking here at cobalt and again at molybdenum, when we look at anthesis versus veraison versus harvest, different seasonal periods, ion concentrations change as well. And they're different based on the root systems to which the Chamberson plant is grafted. And then finally, at the bottom, here we looked at the leaf position. So of course, we're talking about vines. So there are leaves at the tips of vines that are very young. We just use Y for young. There are leaves at the middle of the vine, M for middle, and then there are the older leaves that are the first leaves that were made, but are the oldest on a given shoot. And the elemental concentration of those leaves vary based on their position. So when we think about a perennial plant phenotype, it's really quite complex, and all of the complexities of these different aspects of the phenotype change a bit based on what the rootstock is. So I'll just give a couple of other examples. Zach went on to analyze our transcriptomic data. These are just weighted gene co-expression network analyses that show that different gene regions vary in their expression in the scion based on the rootstock to which they're grafted. So each one of these 4 points is a Chamberson transcriptome with a slightly different rootstock. And then lastly, there was another wonderful graduate student, Bridget Williams, who was really interested in epigenetics. And something we've been thinking a lot about in perennial long-lived plants is whether they bear a signature in their epigenome of the environments that they are experiencing. And there's some hypotheses that this may be a means for clonally propagated plants to adapt, not in the evolutionary sense of the word, but in terms of dealing with their surroundings through this mechanism. So what Bridget did is she took that same experimental vineyard that I described, and she looked at two axes of variation. One was she looked at vines that were ungrafted. Again, these are all Chamberson, and some of those Chamberson were growing on their own roots. Vines that were ungrafted, and compared them to vines that were grafted. So we're comparing the triangles to the circles. She also then compared ungrafted, okay, I'm sorry, I got this a little bit backwards. She looked at the ungrafted and the grafted, and for each of those, there were irrigated, shown in dark blue, and unirrigated, shown in light blue, vines. So she went through and did whole genome bisulfite sequencing to ask the question whether these treatments affected the epigenome of the leaves. And the short answer is that they did. So this principal components analysis shows, you can see our triangles up here at the top and our circles here at the bottom. What these show is that plants that are ungrafted, and with the triangles, differ in their epigenetic signature from the plants that were grafted. Now, also of interest is that those ungrafted plants, we saw the irrigated, shown in dark blue, separating from the unirrigated, shown in light blue. But that difference was not observed in the plants that were grafted. So here we see differences in epigenetic signatures in the grafted scion, all Chamberson genetically, due to grafting, and then differential responses to irrigation, epigenetic responses to irrigation. This really piqued our interest. And it got us thinking maybe there's this whole other dimension of the perennial plant phenotype that we really just haven't been paying enough attention to. So we took these and other data and we planned the next series of projects in grapevines, and this is where we are right now. So we're trying to deconstruct this phenotypic plasticity that we observe in our clonally propagated grafted grapevines, both in terms of the genetics and the epigenetic architecture of the scion and the rootstock. So we have 2 vineyards based in St. Louis right now. These vineyards are replicated across our collaborator sites in South Dakota, New York, and Pennsylvania. One of the vineyards, we have a rootstock mapping population. So we've gone ahead and made an F1 cross of rootstocks. Each rootstock is genetically different from one another, which is separate from the previous study that we did, and then grafted a common scion to that rootstock mapping population. So we're trying to map the genetic effects in the rootstock on the phenotypic effect on the scion. And this vineyard is just coming into fruiting now, so our current work is focused on berries and berry chemistry. The other vineyard that we just set up this spring is what we're calling an epigenomics vineyard. This adorable little baby vineyard has 4 different scions or cultivated varieties that are either grafted to themselves or grafted to 4 different commercial rootstocks. We sourced this vineyard from the same location, and we planted it in St. Louis and in South Dakota, New York, and Pennsylvania. And what we'll be doing is looking for epigenetic accumulation over time. So we took data for the baseline this fall, and then over the next 4 years, we'll be collecting both phenotypic but also epigenetic data. There's just a world of work to be done in systems like grapevines, and this is not the only one. Most perennial woody crops are clonally propagated, but this has been the one that's drawn the most attention, that's been the easiest to fund, and has presented a lot of opportunities for understanding just how plants are doing what they're doing despite being clonally propagated and growing over multiple years. All right, so I want to just shift gears a bit. We're going to be leaving the world of grapevines and wine for a moment and heading into more perennial herbaceous species. That last vignette focused on the organism. Moving into the next section of the talk, we'll be moving a bit into organs and tissues and also a bit into different species and different communities. So I'd like to move now to ask this question: how do traits co-vary among organ systems? Does this change over temporal scales, among species, or within communities? Now, this is work that really started through our ongoing collaboration with the Land Institute in Salina, Kansas. Is anyone here familiar with the Land Institute? Wonderful. If you aren't, I highly recommend you take a look at it. This is a nonprofit, 40 years old, well, maybe 45 years old now, maybe a little older than that, that has been advancing perennial polyculture agriculture. We've been working with them since 2014. They've funded some of this work, as has the NSF Biology Integration Institute that Amy mentioned. So I want to first, for those of you that might not be familiar with the concept of perennial herbaceous alternatives to major grains and legumes, this can be a bit of like a jarring concept if you haven't heard about it. The issue is that most of our grain agriculture comes from, for millennia, comes from short-lived annual plants that are now increasingly grown in monocultures. And while these are highly productive, we know that there are issues with ecosystem services and impacts on the soil. So there are folks who are moving to try to develop other types of grain crops. These were never domesticated by early farmers. So it's a really recent shift into really domesticating, breeding perennial grain crops. And the one that's the furthest along is this perennial grain crop called intermediate wheatgrass. The trade name is Kernza. It's Thinopyrum intermedium. It's a relative of wheat. It makes a grain that can be ground into flour. You can buy that flour on Amazon. You can buy it through a company called Sustainagrain. You can pretty much bake with it. I use it just as a substitute for regular flour. I usually just use half Kernza, half regular flour. Make some coffee cake or a Dutch pancake or some chocolate chip cookies. Folks are brewing beer with it. It's an actual usable emerging perennial grain crop. And it's one of many that we have been studying for the last almost 10 years. So in trying to facilitate this shift from annual crops to perennial crops, and in trying to build a botanical foundation for how domestication that's focused really mostly on yield might also attempt to preserve, if not optimize, the root system, we wanted to do some basic biology. And we just wanted to study how these plants grow and how they're allocating resources over time. So I want to walk through the setup for this experiment first, and then I'll focus most of what I'm going to share today on the root system. So we started with 13 perennial species from 3 plant families: the sunflower family, the bean family, and the grass family. We have Kernza in here, Thinopyrum intermedium. We have another perennial oil seed that the Land Institute's developing, Silphium integrifolium. This is a native North American species. And then we have a bunch of other stuff. How did we pick these species? Well, either we knew them, we liked them, they look kind of croppy. This is a relative of barley. They grew well in St. Louis. Others grew less well, like Lupinus polyphyllus. We just got started. And the first question we wanted to ask was, okay, how do traits expressed within the lifespan of a perennial plant relate to one another? So we started with seeds in 2019 and in 2020, and we began phenotyping seeds: size, shape, weight. We quantified germination date, timing to germination, and then we took all these individuals we had these 13 species, and we literally had hundreds of seeds per species. We began to capture some early growth using a phenotyping system at the Danforth Center. We tracked these plants by individuals, and then ultimately we planted them out in the field where we monitored them over the course of 3 years. This figure only goes through 2023, but many of the plants were in the field past that. So I wanted to share one of the cool tools that we have at the Danforth Center, which is an automated phenotyping machine. Let me just get this going. Whoop, hold on, it's a little sensitive here. Okay, so basically this is a growth chamber that holds 1,140 plants. Every plant is barcoded, labeled, and is watered mechanically at a regular rate every day. And what we did, I'm super proud to say my lab group doubled the number of species that had ever been on this machine with our first experiment. But the reason that we use this is there's both an RGB camera and a near-infrared camera that takes pictures of these plants every day from the top down and from the side. And that allows us to quantify growth rate over about a 6-week period. So after we collected the seed phenotypic data and some of the other early life stage germination data, we put the plants on this machine for 6 weeks. Some of them got so big that they were just very bad tenants in this whole system. They toppled over, the technicians would get called in the middle of the night, middle of the weekend. It wasn't damaged, the machine was not damaged, but it did definitely tested the limits. After we pulled the plants off this machine, we went ahead and threw them into a U-Haul and you hauled them out to the Shaw Nature Reserve, where we built about 1/3 of an acre experiment with landscape fabric and planted all these plants in the ground. And that's where we collected our phenotypic data. The phenotypes we were most interested in the ground were flowering time, height, and then yield. So this is a piece of the data that I'm not going to show today, because I want to spend most of the time talking about the really interesting part, which is what was going on below ground. So Matthew Rubin, who was a research scientist in my lab group for 5 years, just started a faculty position at St. Louis University, designed this experiment, set it up, and collected all the above ground data. And then we hired Molly Hanlon, who is a senior research scientist. She's a root biologist who was tasked with trying to figure out what was going on below ground in all of these species, which was no easy feat. So what we were asking is, how do shoot and root traits covary on the organismal level, the species level, the family level, and beyond? So we had all the above ground phenotypes, both early life stage and later life stage, already recorded. Molly came in and taught us how to take soil cores. This is something we'd never done in my lab group, which we did over the course of 2 years. And then we destructively harvested, essentially dug up a subset of the species, the ones that are shown here. So I want to tell you a little bit about what we've learned about what's going on above and below ground in these perennial herbaceous species. And I'll be focusing again on this Thinopyrum intermedium. This is this perennial wheat alternative, Kernza, as well as this emerging perennial oilseed, Silphium integrifolium. We're going to start here with Silphium. Okay, this is what it looks like to try to soil core perennial herbaceous species and actually dig them up. It's back-breaking labor that was done a little bit by me, but very little, mostly by a huge army of students, both graduate and undergraduates, technicians, and really kind people who were willing to help us out. Okay, so the first thing that we started looking at were universal traits like root depth. I mean, it's not that easy to know, like, how deep do the roots go? And we weren't able to get all the way to their end, but what we were able to observe were both inter and intra-specific variation in root depth. So these figures, which Molly put together, show root biomass by depth. And you can see that some of the grasses, this is Canadian wild rye and some other relatives, blue rye, Elymus canadensis, Glaucus, Virginicus, Thinopyrum intermedium. Mostly these guys have most of their below ground biomass closer to the surface. Whereas some of our other plants, including Tripsicum dactyloides, that's an eastern gamma grass, Silphium integrifolium, have a little bit more of their biomass located slightly deeper. Just getting a window of where roots are was what we were hoping to start with. So let's dig a little deeper on Silphium here. So one of the nice things about our Silphium experiment is when we cored and when we dug the plants up, we had plants that we had put in different years, and as a result, they were different ages. So here we're comparing total root length, which, just as a note, is the total length of the roots in a soil core, but it's not actually itself a measure of root depth. And we're looking at the plant age, whether they were 2 years old, 3 years old, or 4 years old. And what we're seeing is that total root length increases with the age of the plant. And maybe this is groundbreaking, not to make a bad pun, maybe it's not, but above ground, what we're seeing is plants, making structures, dying back over the winter, and making new structures. Those plants are not necessarily getting bigger over time, but their root systems are, and that was important for us to know. We also saw this difference in deep root length going from younger plants to older plants. But when we looked at the D75, which is a measure of where the roots are in the soil core, we didn't see as much of a difference over time. So although these plants are making more roots, they're not necessarily making deeper roots over the course of their lifespan. Now, Silphium offers another nice opportunity because we had planted within our experiment wild Silphium, which are shown in purple, but also Silphium that had been improved through selection for increased yield and increased feminization. These are targets of selection for this plant. And what we're seeing with, well, and then there's also a hybrid, a wild by improved F1. And what we're seeing, again, when we look at total root length, is that the improved plants are showing increased root length relative to the wild. Same for this measure of deep root length, but not for, again, this measure of D75, the distribution of roots within the soil. So one of the fundamental questions in perennial grain development is, if you select for increased yield, which all breeders need to do, are you also selecting for increased or decreased root mass? And what we want is we want to be able to develop crops that both produce at the level we need, but also retain or enhance those critical below ground structures and functions. Okay, so a couple other things. Molly set up this really nice UV light anatomy microscopy within the lab so that we could begin to look at some of the structures of the roots. I'm not going to show those data today. I'll just, again, focus on the relationship between specific root length, which is root length per mass, juxtaposed against shoot dry biomass. So one of the questions, again, we're trying to get at is, how do lower levels of organization link to plant performance above ground and beyond? So here we can ask how specific root length varies versus shoot dry biomass. So these are dicots, so they have thick roots that are beginning to accumulate some secondary growth, roots of medium thickness, and then roots of that are much thinner. And in looking at this, in looking at this first figure, these axes have been normalized here, but if we release those axes and are able to see the differences between thick roots, medium roots, and thin roots, we see a difference between we see a relationship, an inverse relationship between specific root length and shoot dry biomass. So roots that have higher specific root length, these are roots that are often classified as do-it-yourself plants. They're exploring a lot of the soil at relatively low cost. I think of these as kind of thinner and longer roots. That just helps in my own thinking. I don't know if a root biologist would describe it quite in that way. But the higher the SRL, the more explorative the roots are. And here we're seeing higher SRL is associated with lower shoot dry biomass. So we're seeing that really bigger plants above ground that have higher shoot dry biomass are relatively have less acquisitive do-it-yourself root systems. So the last thing we can look at then, or that I'll show here, is we again have this comparison of improved lines shown in yellow, wild lines shown in purple, and then the hybrid shown in gray. And although we see that relationship in between specific root length and shoot dry biomass in the wild, it disappears in the improved lines. So it does appear that there is some shift in root systems that's happening in association with selection for increased yield. Okay, the last thing that I want to mention in this section of, I think this is almost the second to last thing, is the question of whether plants with increased above ground biomass have increased weight of everything, and whether that is associated with deeper rooting. So here we're going to shift to IWG, or intermediate wheatgrass. Again, this is this Kernza. We have data both from above ground and from below ground, and we're just looking at the relationship between rhizome weight and shoot weight, root weight and shoot weight, yield and shoot weight, stem count, shoot weight, rhizome weight and root weight, and yield and rhizome weight. And what you can see, and is said here in the title, is that plants that have increased biomass above ground also seem to have more structures below ground. What's less clear is where those structures actually are in the root column. So just like with Silphium, with this intermediate wheatgrass or Kernza, we had plants that were planted in 2019 and those that were planted in 2020. So the 2019 plants shown in red, these are the older plants. The 2020 plants are shown in blue, are the younger plants. Again, we harvested these at the same time. So here we're showing that the older plants at the time of harvest have less shoot biomass and lower plant height than the younger plants. So they grew big and then they grew less big over the course of their lifespan. But when we look at the roots, we see the opposite pattern, where the older plants, the red plants planted in 2019, have greater total root length and actually deeper root length than the younger plants that were planted in 2020. So although we're seeing over time a decrease in shoot biomass, we're seeing the opposite in the roots of this perennial grain alternative. So this, again, I think argues for maybe the potential of a perennial system where root mass of plants that are left to grow increase over time. The trick will be whether we can maximize that and yield. All right, so this is actually the last experiment I wanted to talk about, is what happens when we take those plants and we put them in different types of communities. So how does root shoot plant trait variation differ whether you're in a monoculture or you're in a biculture? So we have a multi-site experiment going at the Danforth Center's field research site. This is just a little north and west of St. Louis, where we have intermediate wheatgrass or kernza growing by itself. And the monocultures are both unfertilized, and then there's a fertilized treatment. We then have that intermediate wheatgrass or kernza growing in a biculture with alfalfa, as shown here. And then we have alfalfa growing by itself. 4 treatments, replicated 4 times. I'm going to talk about data from 2 sites within St. Louis that are about a half an hour away from each other. This experiment's replicated twice in St. Louis. It's also replicated in 5 other locations from Kansas to Vermont, but I'm not going to show those data today. And what I wanted to just talk about quickly, we don't have the root data yet available, but we do have the intermediate wheatgrass biomass, which is shown on the top, and the grain yield, which is shown on the bottom. So let me orient you a bit. The top is biomass, the bottom is grain yield. We have 2 sites. One is called Plant Haven. The second is called the Danforth Center Field Research Site. And we're looking at 3 treatments shown here. Intermediate wheatgrass grown by itself with no nitrogen treatment in red. Intermediate wheatgrass grown by itself with a nitrogen treatment shown in green. And intermediate wheatgrass grown in a biculture with alfalfa. These data were analyzed by Kenneth Okonkwo, who is a graduate student in the lab. So what's interesting is, first of all, there's a difference in the sites. So at one of the sites, Plant Haven, we see no differences in either biomass or grain yield based on whether the plants are in monoculture or in biculture. The second site, the field research site, we actually do see differences both in biomass and in grain yield. Next thing I want to point out is in this Danforth Center field research site, we see increased biomass and increased grain yield, both across 2 years in the intermediate wheatgrass or kernza that's grown in a biculture. So one of the questions that folks are curious about is, can we get away from nitrogen amendments by planting with a legume? This is an old idea, but folks have been quantifying it in various studies of late. And what this study shows and suggests for this particular site is that actually, yes, we do see increased biomass and yield in bicultures versus monocultures, regardless of whether nitrogen is applied. Okay, we only saw it in one site, and we'll see what we see across these other 5 sites. But that's where the data are today, and the root data, stay tuned. All right, so where are we? We've been looking at these broader concepts of capturing the perennial plant phenotype. We started with a single organism, root shoot in grapevines. We moved into a suite of perennial herbaceous species where we looked at organs and tissues, and we started to look across a few different species. Now in this last part of the talk, what I'd like to do is move a little bit further out towards our more ecosystem questions and share a new, what are we going to call it, like a new trajectory in my lab. I'm a little nervous to share this, but I'm excited to see what you have to say. So here I want to tell you about our attempts to begin asking how perennial plant traits are influencing soil structure, composition, and gas flux. We're very early on, again, in this work. This is being spearheaded by a terrific postdoc, Stella Wolchin, recent USDA postdoc fellow, who started something last year called Project Trench. And when she came to me with this idea, I was like, what? A bulldozer? Okay, all right, let's see. And we took some of our experiments at the field research site and we dug a trench. And what Stella was interested in doing for 2 different species was asking, okay, we know the root systems of intermediate wheatgrass, Kernza, and Silphium are like super different. We have dried plants in our lab and they're like unrecognizable in terms of their dissimilarities in root systems. But what does that actually matter for the soil? So the trench was dug for the purposes of seeing if we could detect any species-specific soil effects. And what Stella did, she spent maybe about a month in this trench collecting a range of different samples. So she took soil cores from each horizon, measured bulk density, gravimetric water content, total root length, mean root diameter, pore size using a high prop. She studied soil gas, both at the surface and in each horizon. She took soil cores to look at horizonation, soil texture, bulk density, and associated properties, root traits, and then also took sections for XRT imaging, and then lastly collected some samples for root and soil analyses, microbiome analyses. So I just want to share a few very preliminary data slides that have caught my attention. So soil gas fluxes. In looking at Kernza shown in red, Silphium shown in blue, there are relatively few differences. So Stella put together, she knows I'm not a soil scientist, so she put together this really nice description. What are we actually looking at? Soil gas flux, the exchange of these gases between the atmosphere and soil at the soil surface. No differences. So then the next thing she did was to look at the soil gas profile. So here, Thinopyrum intermedium is shown in blue, Silphium integrifolium is shown in yellow. What we're looking at here is the soil gas concentration within the profile that represents gases that are actually stored in the soil pore space. So carbon dioxide concentration, nitrous oxide, and methane. And then looking at the soil depth range, so going from the top slightly deeper, what stands out to me is that there's some differences in the soil gas profile based on the species that's growing in that area. Maybe this is expected, but again, I'm a newbie in this space, so it caught my eye. And then the last thing that I wanted to show here is the bulk density and the total porosity. So here, bulk density, a measure of soil compaction, porosity calculated from bulk density, the percent of a volume that's occupied by pore space rather than mineral particles. And again, we're starting to see signals of some differences, particularly in the higher soil horizons based on the species that are planted there. All right, so this is really, again, just the very beginning of our foray into asking the question, is there a plant species or plant genotype specific effect on the soil? And really the reason we're asking this is we're wondering, can we optimize breeding? Can we optimize selection for roots that will have the desired effects in the soil? Because when we think about perennial polyculture agriculture, we're thinking not just about what's going on above ground, but we're really talking about optimizing for ecosystem services. All right, so what we've done so far is to look at biological foundations of the perennial plant phenotype. We used grapes as an experimental system for how we can understand how roots affect shoots. We then moved into a suite of different perennial herbaceous species where we studied relationships among traits above ground and below ground. And then lastly, took a look at how some perennial plant traits, in this case, what I showed today, just the traits of 2 very different species, may influence soil structure, composition, and gas flux. So in the last like 3 minutes, what I want to do is just bring it to some breeding applications. So the vision here is to lean on the example that nature has provided in perenniality and diversity and try to adapt that in some meaningful way in agriculture, which is at the moment looking like not a hugely diverse mixture, but maybe something as simple as a biculture, where you have 2 or maybe a few more species growing and rotating in rows. Again, what we're trying to incorporate into agriculture is this incredible below-ground biodiversity and functionality that we see in nature. So the last thing I wanted to share is that we've actually been trying to expedite and optimize the breeding process, really reimagining how we develop crops for agriculture using something called a phenomic prediction pipeline. And what this is it kind of builds on that experiment that I shared where we had the spaced plant nursery of 13 species. And what that experiment showed is that there are clear relationships between seeds and seedlings and traits that we see later in the field. And so the vision that we had with our collaborators at the Land Institute was, can we predict, based on seed and seedling data, what plants will do later in the field? This is an old idea called early stage selection. Our new take on it is that we're using hyperspectral data of seeds and seedlings as a multidimensional data set to do that. So this is work that's funded by the Foundation for Food and Agriculture Research, or FAR. We're working in 3 species that you've seen 2 of which already: intermediate wheatgrass, or Kernza, silphium, also known as silflower, and then a legume named sainfoin. We collected various spectral data on seeds and seedlings over the course of multiple cohorts that went out into the field at multiple years. We're using a range of different devices to collect spectral signatures. These are handheld spectrometers that record wavelengths at different parts of the spectrum. And then we went ahead, once we collected all of those data, we took the same plants, put them out in the field, and then have been collecting various in-field traits: overwinter survival, plant size, flowering time, and yield across 2 sites across 3 years. And these are the data. It takes a long time. Like, I look at this, I think, man, 2021, that's a long time ago. But it takes a while to let these plants grow and for us to get the data needed to develop the actual models. And I just want to give one example and say, in silphium, there are these ligules, or the rays of the ray florets. And ligule number, we know, is a proxy for seed production, because in this species, only the ray florets are bisexual. They're the only ones that make seeds. And so we wanted to ask the question, could we predict ligule number using this phenomic prediction pipeline based on spectral data taken on seeds and seedlings? And so we generated a relationship matrix based on these high-dimensional seed and seedling spectral data. We evaluated more than 100 combinations of different high-dimensional seed and seedling data sets. So I mentioned we took spectra using different handheld spectrometers. We took them in different times, different species. And then we trained models for ligule number using silphium plantings, what we called training sets, in 2 sites. One was in St. Louis and one was in Kansas. We were unable to collect data in a 3rd training set because the plot was inadvertently bowed down. And then we used those models to predict ligule number in a novel environment, a test set, based on relationships that we established in the training set. So the reason I wanted to be sure I get this in today is what we're finding is that the relationship matrices constructed using these training sets can actually predict ligule number. So our preliminary models show correlations between ligule number predicted based on the relationship matrix and numbers of the ligules observed in the test set. And those correlations range between 0.25 and 0.45 depending on the model, which is significantly greater than random. So what we have here, we think, is a mechanism where we can, through seed and seedling selection, predict traits that we see later in the field. And our big picture vision would be to apply this approach for a range of different traits. So I gave ligule count as the example, but we've actually looked at this in a number of different traits, including seed weight, head number, number of disease traits, blotch and rust. And we're seeing that this is actually a viable means of prediction. And if we can use this pipeline to predict ligule count and yield, maybe we can also eventually use it for root traits. Maybe we can even use it for soil properties. All right, so let me back my way out of this talk and wrap things up. What I am most interested in is just like understanding how these beautiful perennial plants grow and allocate resources, and how we can use that understanding to improve our agricultural systems, which are highly productive, but ecologically quite, well, I mean, they're just not great in some areas. So really, like, the take-home dream of my lab group is this concept of like these perennial possibilities. And I think through our work with grapes, I hope we'll be able to adapt and make our woody crops more resilient, maybe through rootstock breeding or something like that. I think we can breed for dual functionality, yield and ecosystem services, and emerging perennial grain crops. I think we can improve how we're running our breeding pipelines in ways that are quicker and cheaper and lend themselves to a broader diversity of species. And lastly, I didn't have a chance to talk about it, but at the core of all of this work is the conservation and accessibility of crop genetic resources of seeds. This is the seed storage facility in Fort Collins, Colorado. And this is something that I think requires attention and support, and it's kind of concerning at the current moment. All right, so I'll just end by thanking the members of my lab group. We have a diverse group of students from St. Louis University and technicians at the Danforth Center who are incredibly hardworking, wonderful people. I'm a lucky person to have them on my team. And then our New Roots for Restoration Biology Integration Institute that's facilitated a lot of this work. And with that, I'll stop and take any questions. Thank you. Yes.
All right.
Oh, sorry, Amy.
Allison, oh, so yeah, thanks very much, Allison, and I'll let you take questions.
Yeah.
Awesome talk. I learned so much. I'm wondering how much does phenotypic variation, how much does it explain epigenetic changes versus genetic?
Okay, so yeah, the question is how much of phenotypic plasticity is due to epigenetics versus genetics? Amazing question. I don't know the answer. I hope we'll learn more about this with our epigenetics study. We do have 2 genetics studies underway, aside from the grapevine one that I talked about, where we have a F1 backcross population between 2 silphium species, where we've been collecting above ground and below ground data. I hope that study will tell us a little bit about the heritability of some of the traits that we're looking at. And I hope it will tell us a little bit about how, the extent of plasticity and whether there is a genetic basis to that. But we just got the map back, and we're just doing those early analyses. But I don't know. I mean, I think there's a lot we don't know about epigenetics. And I wish if I wasn't a soil scientist, I think I'd be an epigenetics biologist. So maybe in my next life. Yeah, great question. Yeah.
Thanks so much, Allison. The one thing that really stood out to me was your results with the alfalfa versus nitrogen additions, and so those were like strong enhancements, and it wasn't seemingly a huge response to nitrogen. Do you think there's something else going on in there? And also, side question on that, did you look at forage quality? Like, are you seeing biomass? But what does that mean for quality?
Yeah, really good question. So I'll paraphrase by saying like what.
And make sure to say what her question was.
Yeah, well, I will paraphrase to the best of my ability here. In the experiment in which we have intermediate wheatgrass growing in a biculture with alfalfa by itself with nitrogen, what do we think is going on, basically? And is there a forage quality effect? Excellent question. There's actually several groups that have been looking at this particular experiment. So we are 100% not the first people to do this. The results are consistent that we are seeing this increased yield, increased biomass, more or less in biculture versus the nitrogen amendment. And we are seeing it across sites. although in the example that we had where you have 2 local sites, it's not always the same. And honestly, our local site is, one of them is slightly better than the other in terms of soil quality, et cetera. So that's a whole other thing that we should probably explore. I think what I hope is that the alfalfa is providing the more than enough nitrogen, and that's what we're seeing. When we compare alfalfa in biculture to alfalfa by itself, the alfalfa in biculture is a lot smaller. Like, it's definitely making nitrogen that it is giving away to the benefit of one and not the other. As far as forage quality, we haven't looked at that, but we harvest alfalfa 3 times a year. We harvest the kernza just once. There's a lot of biomass coming off, and what's in that biomass, I'm like kicking myself now that we didn't think to do that, but we didn't. But it's not too late. Maybe next year we can incorporate that. But yeah, I don't know. But great question. Should be done. And maybe somebody knows, not me, and I would have to check the literature. But yeah. Yeah.
Yeah, great talk, thank you. I was curious about the early chart you showed, the principal components analysis on grafting versus irrigation. Do you have a sense of what those splits were, especially those for the grafted that split apart with irrigation?
Right.
What were the variables?
Yeah, go ahead.
I'm curious about the variables, if you had some functional traits that you could assign to the split, what was actually causing those splits?
Yeah, let's, maybe I'll just zoom back there really quickly if I can. Oh gosh, I don't know if I can do it easily. I think it has to do with the physical trauma of grafting, long story short. So when you look at X-ray images of what a graft junction looks like, it's a very valiant, but in some cases sad attempt of a plant to try to reconnect its tissues. And I think there is a wound response that happens, and I think there is a physical trauma that happens, and I think that affects how a plant functions. That would be my guess as to why we're seeing that. Now, how that affects epigenetic modification and how it affects the movement of molecules, small RNAs, for example, from the root to the shoot, presumably, I would assume that there is an effect of that. I don't know what it is, but here in the state of New York, I think is the best person that I know of working on this, which is Margaret Frank, who's at Cornell, who's been studying the effects of grafting on various trafficking of molecules. I think she's been doing it mostly in tomato. But I think her work would be more insightful to that question than ours is. So I'll point you to Margaret. Yeah.
All right, well, why don't we continue on with questions in the lunchroom with Allison, so we can move over there for lunch. And then at 4:30, she'll be back in the lunchroom. And so if you want some group chat Q&A with her then, we'll also be available then. So let's thank Allison for an awesome talk today.